Hello, good afternoon everyone, and a very warm welcome to you all. My name is Katie Williams and I'm the Technical and Product Development Manager here at Denji. Our webinar this afternoon takes us away from our sort of core territory of horses really, and producing horse feed with all the issues that entails. And we're going to enter the wonderful world of what I usually refer to as exotic animals. To do that, we have two very special guests with us this afternoon, Tullis Matson and his team, I hasten to add, at Nature's Safe, as well as Andy Beer of Zoocraft Nutrition. I'm going to introduce them more fully in just a minute, and I feel a bit rude doing this, but I'm going to just set the scene for today's webinar with a few slides about Denji. So just bear with me if I can move this along. There we go. So this is Denji down here in Essex, with basically a very sort of rural company based out in East Essex. We're about the last thing you come to before you hit the sea. And it is a farmer's own business. So we started over 50 years ago with a bare field site, and since then have expanded to take up quite a large proportion of the village of Ashildon. And you can see that we have a lot of grain silos as well, which are basically another part of the feeds business that we run. But most of what we do is about growing lucerne or alfalfa, and I've already used the two different names for the crop, which I've put on this slide because we do use them interchangeably and it can cause a bit of confusion, but they are two names for the same basic crop. We are the UK's largest grower of alfalfa, so we have around 5,000 acres down here in Essex and then a further 1,000 acres in Lincolnshire, which is owned by a company that we have a partnership agreement with. Everything that we grow in Essex is within around a 15-mile radius of that site that you've just seen, and that enables us to keep our feed miles very low. So yes, we do import some ingredients from around the world, but it's less than about 5% of what we use in total. So as a company, we have very low feed miles. And as I mentioned, we are a farmer-owned business, and everything we do is really with the intention of adding value to what they do. We've been around for 50 years and we want to be around for at least another 50 years. Alfalfa makes a great crop for them. So their cereal growing is what they primarily do. And then the alfalfa is used to put sort of nitrogen and things back into the soil. So it is a very useful crop in that sense. Alfalfa itself is actually quite tolerant of the UK weather, so it does survive droughts. Obviously the most extreme conditions we've had probably in the recent sort of 3 or 4 years, not least this summer, have tested its resilience, but it is tolerant and it does keep going and it will come back. So although yields might be reduced, it does survive. We typically cut 3 times a year on a roughly 6 to 8 week cycle, depending on weather, and we would rotate the fields every 3 years. So as you can see in some of these images, we are left with a stubble over winter, so it provides a bit of ground cover for various animals and insects, and it also does help to reduce a bit of soil erosion as well by having that stubble in the winter months. The lattice heath moth that you can see there in the middle picture absolutely adores alfalfa. The flowers are a magnet to it, so again we're helping a lot of wildlife and diversity in what could be an area that would be vulnerable to sort of monoculture cereal growing. As I mentioned, our farmers usually follow the alfalfa with wheat to take advantage of the nitrogen, so it means that they're putting less sort of nitrogenous fertiliser back in, and as I'm sure you're all aware, the prices of fertiliser have been through the roof in recent years, so it is a very advantageous crop in that sense, and increasingly so. And we call it precision drying these days because the temperatures have been reduced gradually over the last 20 years, but effectively we're tumble drying the crop. So we use hot air to take the moisture out, and that bottom image shows the moisture. It's not sort of smoke or pollution, shall we say. It is the steam that goes off into the environment, the moisture that we take out the crop by using hot air and tumble drying it. So it's producing a consistently clean forage from bag to bag. So it is an intensive process, but it traps all the nutrients in, and that's what obviously animals eating the alfalfa benefit from. And I think key, and just to round off really, we are keen to remain a sustainable business. We've been here for 50 years. We want to be here for another 50 years. We're on our third generation of farmer members that are on our board. We have a royal warrant, which reflects our commitment to our environmental and social responsibilities. And we continue to work to reduce our energy use, managing our packaging materials better, and trying to improve our manufacturing efficiency as well. And not least, as part of our commitment to a more sustainable future, we are trying to support organisations like the Rare Breed Survival Trust, Essex Wildlife Trust, which is very local to us here, and organisations like Nature Safe, which we're going to hear a lot more about from Tullis in just a minute. So it's a very brief overview, because I realise that as a host to hog the limelight, it's a very rude thing to do, but it really is designed to give you a bit of background as to why we wanted to organise this webinar, why it fits with the Dengie ethos of trying to support a range of different animals with the best nutrition, and to support those who really want to make a difference to the planet and the flora and fauna that we share it with. Which leads me very nicely on to our guest speakers this afternoon. I'm going to start with Tullis. So if I stop sharing my screen, he's going to appear on the screen as if by magic. Tullis needs very little introduction really to those in the horse industry as the man behind Stallion AI, but you may not know he began his career with horses working in a racing stables as assistant trainer to Philip Hobbs. In the 90s he came home to run his father's non-thoroughbred stud at Twemlows Hall, and also at that time toured New Zealand to learn more about AI and reproductive technology. He then returned home to begin practicing AI for the first time, and during those intervening years, Stallion AI Services has been running. Tullis has frozen semen from some of the world's greatest stallions, and I would like to add some of the rarest breeds of horses, and as if by magic, Tullis preempted this and has a breed very close to my heart, which is the Suffolk Punch on the screen, and hence how the connection really began between our two organisations. In 2011, Tullis received the Marsh Christian Award for Conservation in Genetic Biodiversity, and I know he's going to tell us a lot more, and we're going to be treated to a very special behind the scenes tour of his latest venture, which is Nature Safe. So over to you, Tullis, and the team there at Nature Safe.
I think that's better. You can hear me now, can you, Katie?
Yeah, perfect. Thanks, Tullis.
Right, sorry, yeah, it's not me myself. So thank you, Katie, for that lovely introduction and presentation as well. And thank you, Dengie, for supporting Nature Safe in everything you do. Really, really appreciate that. I mean, it's quite interesting. Obviously, right at the beginning, you said, obviously, you're very much around the horse world and what you do. And I know you're going to the exotic side, but I thought we'd start off with a horse. It's a very, very special horse and a very, very special lady. And I know you said it's quite dear to your heart as well, and they're very dear to our hearts as well. So this is a Suffolk Punch. There's about 80 to 90 breeding females left in the country, 300 or 400 in the world. They're just an exceptionally amazing animal. They really are. They're the ones that ploughed the fields in the 1940s. I've got a picture of my grandfather actually out in his fields, ploughing the fields. So, and as it's not so much a use for them now, they sort of declined in numbers, and it's very, very sad. They're part of our heritage. You know, they put food on our table during the war, and I think it's very sad to see them disappear because they're such an amazing animal as well. So we've always been trying to find ways that we can use new technologies to try and save our rare breeds. I was very, very fortunate to be a trustee of the Rare Breed Survival Trust, which I see you support as well, which is fantastic. It's really important to take care of our heritage. And we looked into there, seeing how can we help these rare breeds of horses. And I love the heavy horses themselves. And one of the ways we were looking at biobanking, tissue banking, which we're going to learn an awful lot more about this later, and seeing if we can take a bit of tissue, but also freeze the semen down from these animals as well, and preserve that in time, because you never know when you're going to be able to need it in the future. And I thought, well, if we can do this to this amazing animal here, why can't we do it on a rhino or elephant or something like that? And that's what we're going to lead on to with Nature Safe. That's how Nature Safe was sort of born. About 6 years ago, we thought, well, yeah, if we can try and save this amazing animal, this breed, why can't we save other species as well? And that's where the sort of the idea came from originally. So yeah, we had the first foal on the ground for a rare breed by sex semen as well. So we're using all new technologies, and we're going to talk all about how these technologies out there and what Denji is doing to sort of supporting Nature Safe, and how that is actually helping save these amazing species. So we better say farewell to Diamond. She's one of our teams in there. And Millie, of course, another special lady. We mustn't forget Millie as well. Who's patiently there feeding polos or mints.
Suffolk will do anything for a polo.
Yeah, so, but they are. Katie, you say you've got a bit of an affinity with Suffolk, have you?
Yeah, well, I worked with them for many years, from about the age of 18 onwards, when I moved to Suffolk, actually the county of Suffolk, and we had some trouble breeding with some of the mares. They were not the most fertile, but I think you can trace them back to one stallion, so it's no surprise that there are issues with them, really. Well,
they've got some of these heavy breeds have got a big inbreeding issue, and again we're going to come on to that. And again, that's where Nature Safe plays its part. If we can preserve these genetics down, we can bring them back in the future. But I ask any of your audience out there, if you get a chance to get up close and personal to a Suffolk, I would fully recommend it. As of course any other heavy breeds and rare breeds.
Yes, definitely.
I can't leave the other ones out, can we? That'll be
no. So they'll be struck off the Rare Breed Survival Trust.
I will. But anyway, we'll move on into the lab. So
brilliant, thank you.
Let's say goodbye to her and we'll come on in. So just our stallion board that we have here. So this is our breeding set, just so you know. It's where we collect all the semen. I don't know whether you showed the dummy mare here. This is just where we jump the stallions on and collect the semen off them, and all the stallions up here. I feel a bit sorry for the stallions. They like to have a scoreboard around here. So what the dummy mares' heights like and everything else. So we put through so many, we have to keep an eye on that side. Anyway, come on in. Right. So I've got my daughter behind the camera for the first time, so it is a little bit wobbly. Right, doing a cracking job there, India.
Yeah, it's great, India. Well done.
So this is I'm going to just walk you through to the lab. This is what we call the dirty room, or the AV room. These are the artificial vaginas that we collect off these stallions, and so we have different sizes and shapes for different type of stallions and things like that. That's all done in here. So on this side, this is where all the semen's processed in the lab. So we'll come over here and we'll see how some of this work's doing. Pam, do you want to come here? So this is Pam. Pam's our lab manager, and Pam's been very involved as well with Nature Safe and looking at all different ways we can cryopreserve. We were looking at deer semen the other day, trying to preserve deer. We actually froze down a mouse deer. Believe it or not, mouse deer is the smallest deer in the world. It's about the size of a hare, and it's really, a lot of people might say, why the domestic side and the sort of the exotic side? How can that transfer over? But it does massively. It's really easy for us to collect semen off these other breeds, domestic side, and we learn from that and we transfer that knowledge across. So like we were dealing with the stag the other day, weren't we? And
we looked at it, and the stag and the dog extenders, the different types of extenders, the ways we can process it, all of that, every species we do, we can look at it across a whole and see what we can move over from one species to another.
I feel like sometimes I might be waffling on, we're the luckiest people around doing this job. Well, what were we speaking about this morning on the webinar? On the Zoom? Bees. I mean, it's incredible. Bees and snakes. So how can we preserve bee semen and snake semen? Bees are getting genetically the gene pool's really shrinking, so we've been asked to try and preserve that side of things. So it's really varied when it comes to Nature Safe, what it's doing. But I say it started with the equines, and a lot of the equine side of it we transfer over. So I'll just show over here, actually, if you come over here, it's all about how we freeze these down. And we freeze down, we've got Emma here, who's Actually, Emma, just explain. This isn't, but poor Emma. I didn't tell her we were going to do this, but she's in the lab, so she might as well tell her what are you doing at the moment?
At the moment, we're making two different types of extenders. So we've got equine extender, which is ready to be spun down, and then we've also got some deer extender that we're trying out with just some different sugars and things. So we've got a trial work as well.
So I don't know, you got that. That's all do trial work. So again, it's all transferable over to many of these other species. So sorry, Emma, for dropping you in it there. The last.
Alice, can you just explain what the extender does and why you use it?
Very good point. That is perfect, Katie. Yeah. So basically, when we freeze everything down, and this happens the same when we do a skin cell, for when we're doing the tissue samples. You have a think of what happens in the winter time when your pipes freeze. The water expands, doesn't it? And those pipes burst and they rupture. A sperm cell, a skin cell, an embryo does exactly the same thing. If it's got water in it, it'll just literally rupture and break when you put it into minus 196. So we have to develop an extender or medium, which is this here, which is we call our liquid gold because it's got some secret ingredients in there, but it's got a cryoprotectant, something that freezes it. It's like an antifreeze. So what it does do is it dehydrates that cell. Imagine that's the head of the sperm, dehydrates it, and then it allows us to freeze it. And when we wake it up, thaw it out, it rehydrates. And that is absolutely vital in the freezing process. Now, whether it's a sperm cell or a skin cell, it's exactly the same. Different extenders and mediums we have to use, but it's the same process. Does that make sense, Katie? Does that sort of explain?
It does. And and are you using, did you say, the same extenders for different species, or do you tweak them? Or
no,
are they
very,
completely
very
different?
And that's what I was just doing there. There's so much trial work and experiments going on here all the time. So we've been developing a new freezing medium, haven't we? So
yeah, we've got lots of new media. So if it's the canine, the equine, elephants, rhinos, the one for the stag, and that's the stag have already got one to use, but we think we can make it better. So we're just doing a lot of trial work, changing the sugars. There's all sorts.
And we're even trying to freeze dry it at the moment so we can ship it abroad. So that's something else. So it's add more. So I feel like there's so much trial work that we do here to try and further, but they're similar but different. I know that sounds odd, but they're all very similar components that you have to alter tiny bits of the components within that formula. So most of them are egg-based, but it's the cryoprotectant you put in there, and the percentages are all different. Because the reason for that, Katie, is the permeability of the cell. So some cells are really permeable and some aren't. So some are going to have to take more cryoprotectants. And if you get a cell that's really permeable and put a high percentage of glycerol in it, it's going to damage that sperm cell because it's actually, believe it or not, that the cryoprotectants is quite toxic. So if you put too much in there, it's going to have a negative effect.
Yeah.
Some species you need more. So yeah.
It's interesting stuff, isn't it? Because when you think about the antioxidant protection the body has itself, it's all the vitamins and things that are trying to protect the fatty acid in the cell membranes, and it's the same principle just outside, I guess, and you're having to get that right balance for each species, which is fascinating. No, it is, and it is important. I think we just got some on the screen. I think it's just dying off, so we'll have a look before it dies off too much. But I think this Siemens we just thawed out, and God, I've been doing this for 30 years now, and it amazes me. So, India, can you go up to that screen there? Just go straight up to it.
It's got a bit of drift.
Oh wow, there you go. Brilliant.
So that's so that you can see that on the main screen there on that. Fantastic. We're not. So just check because we're not spotlighted on here. Just try and move that across.
Yeah, you're coming through really. Was that horse, did you say, Tullis, or is that another species? Oh, it's muted. Hang on. Let me see if I can unmute.
Is that better?
Oh, there you go. Oh, no. Sorry. Try that again. I think we've lost the sound. Hang on a second.
There we go. Is that better?
Perfect. Thank you. What have you done? We've got you now. That's good.
That better? Yeah.
Yeah, perfect.
back again. Sorry, I won't touch the button again.
So
But
was
yes,
that
that was 4 semen that were thawed out minus 196 degrees. It's suspended in animation. It's quite incredible how you can freeze it down and bring it back to life in 10 days time, or a year's time, or 1,000 years time. It really doesn't degenerate much at all. So it's quite incredible, that. So, and we've got other devices that we can use now as well. So we can actually come in on here, and we can actually put it on an iPad. We've got something called the iSperm. We can actually analyze the semen on an iPad, and we can literally just press analyze. We go in, and it can actually do it all. So there's lots of different ways we can actually analyze semen now. That's sort of, and this one, I actually took this out when I was out in South Africa, when we collected off 7 bull elephants. We actually, because you can't carry a microscope around, but when we're collecting off these wild animals, it's much easier to take devices like this to get our semen analysis on that side of it. So, right, so that's really just obviously on the really good point about the freezing down, because obviously it follows on from what we're doing. So we have lots of different species through here that we do what's called epididymal sperm extraction. So from the zoo side, we will get a phone call from Chester Zoo or London Zoo and say, look, we've had an animal pass away. We're going to, as long as we get the testes within 24 hours, we come get them here, and we can actually take the semen from the epididymis. It's slightly more trickier than the others. So like a mouse deer, the one we did, the testicle weighed about just over a gram, where the horse's testicle is about 300 grams. So you're dealing with obviously much more detailed anatomy when you're doing that slicing, but it really does work on some of them. I have to admit, a lot of time it fails with some of the more, it's obviously animals, purely because of the fact that no one's ever done this before. And we're actually sort of feeling our way in the dark slightly and thinking what is best. But every time we do one, we learn. Do you know what was the one we did the other day? Was it Hyrax or?
Oh, yeah, the mountain Hyrax, I think.
Mountain Hyrax that we did the other day, which apparently is I don't know much about it. Apparently, it's the closest living relative is the elephant to it. Apparently,
it looks like guinea pig.
It looks like guinea pig. So we dissected those testicles, but that's all relates, and I know that's where our relationship from is obviously Katie from the equine world, and I think we met was it Beaver last year? I can't really remember where we met now.
Yes.
But we transferred all this knowledge from the equines to this. That's where the relativeness is on this. So anyway, we'll move on to the storage. But thanks very much, Kate. A pass. I get that right.
Thank you.
Only been 12 years.
I know you heard it. So I still can't remember your name, but I know. Right. So we'll come on through. So this is, I'll just turn this around. So this is some liquid nitrogen in here. And you can see, so this is minus 100. I'm not going to stick my fingers in here, don't worry, but it's minus 196. Can you see that okay there, Katie?
Yes, steaming in it. Well, is it?
That's it. Yes, well, it's can you see? I don't really see the straws in there. Maybe you can't, but there's some straws sitting in here this morning, and they're frozen down at minus 196. But we're coming into this other room now.
Yeah, don't drop the mobile phone in there, whatever you do. Yeah.
So a lot of these, this is where the storage rooms, this is where a lot of the stuff is stored. So this is like a nuclear bunker in here. So a lot of the genetics are stored in here. And we've got steel doors, concrete roofs and everything else. But I don't know whether you can put your camera in. Well, I don't literally drop my camera, but anyway, you have to push it up or down. I don't know which way. That's it, you got it. So in here, there's about 250,000 straws sitting in this tank. You go up a little bit more. Can you see that there?
Yeah, perfect.
Yeah, so these are all the goblets in here that we've got, and let's say we've got 4 of these tanks, and this is where the genetics, and once they're stored in there, they're held in time. But again, we'll go through more of the science in a minute. So in this tank, if you turn around, go the other way, India. That's it, you got it.
No.
Other way.
There you
Keep
go.
going. Look at it. Right. So in here, a lot of our work's done from the domestic side, from the pet side that we've learned it from sort of the cats, dogs, and horses that preserving their tissue down. But in here is a very special tank. I know we're talking about Nature Safe. Safe stands for saving animals from extinction. And here there are over 230 or 240 different species in this one tank. It's quite incredible. So here we've got things like the southern white rhino, Asian elephants, something like you've never even heard of, the mountain chicken frog. Have you ever heard of that, Katie? And I'm not making no?
Andy might. Andy's nodding. He's heard of it, but he's a zoo expert. I'm not. So
exceptionally rare.
Yeah.
And I think you know, so there's very, very few left in the world. And Andy, yeah, you'll obviously know how rare they are, but we've got about 4 in the tank, the skin samples and parts of the semen. It's quite interesting with the semen. When we got the semen, did the epididymals work on it? It's totally motile. And the way we wake it up is actually adding a bit of drop of water on it, which is a big no-no for everything else. But for the frogs, it works. So there's a whole multitude of species in here. But anyway, we'll go into the other room where you'll meet Lucy, and she can talk about where the processing is. So, right, it's just a walk through the corridor here. Follow me in, yeah?
Yeah, hold on.
That's it. That's it, keep going. Right. And Christmas has arrived already at Studying AI as well. So, I'm pretty sure. Right.
Okay.
Right, so I wanted to introduce you to Lucy.
Lucy.
Lucy's manager of Gemini Genetics. She's also on the scientific board of Nature Safe. So she wears a multitude of hats, but she'll be able to explain an awful lot more about when a sample comes in here from a zoo, what the process is. We're going to go through a PowerPoint as well, a bit more about it, but you can explain a bit about what goes on when they arrive here and what goes on in this room.
Yeah, so when zoo samples arrive, so the foundation of Nature Safe is that we're wanting to bank samples in a way that they can be regenerated in the future. So preserve whatever sample we've got in a way that we can hold it, and then we can wake it back up when we want to use it, and it can still remember and replicate in the future. So when a sample arrives, we work under the biological safety cabinet over here. So it's a bit different to, I think you've been in the Studying AI lab. Ours is a lot more smaller rooms. We need to keep everything sterile. We don't have many people coming through to this lab because we need to keep the environment clean. We work under the hood because we don't want the environment to contaminate the samples. And we receive skin samples plus reproductive organs from the zoo animals. So when we receive skin samples, they go under the hood and they go under a number of sterilisation steps using different media, because we need to be able to control the microorganisms that are on those samples so that they don't interfere with the future use of those tissues. We then preserve them in little cryovials. You can see some there at the back. So it's different to semen, where we're preserving it in straws. In here, we're working with 2 ml cryovials, and we use a special media, and that's really key to what we do at Nature Safe. It's a media that's been developed from the domestic animal work. And that's the thing that enables the skin sample to be preserved down and the DNA able to regenerate once we've preserved it. So after that stage, it goes into liquid nitrogen, a bit like the semen samples. And then when we want to use that tissue, we thaw it out and we begin to culture it. So we can show you here, there's CO2 incubators along here, and I can open the door. In this one, we've got a domestic animal that's in culture at the minute. I can't open the door on that one just because it's quite an important domestic animal.
And what animal's that?
It's the dog. I can show you, this is what a CO2 incubator looks like inside. So we sit a little tray, or we sit 2 trays per animal in this incubator, and it provides a warm, moist, humid environment which encourages the cells to replicate and grow. I can show you just on this microscope here. So this is the sample that we've got in culture at the minute of the domestic animal. I checked it this morning and I've just kept it frozen on the screen ever since. So this culture is in a very primary stage, very, very early on. So you can only just about see, if you kind of know what you're looking for, the cells beginning to attach to the bottom of the flask. So there's 2 skin cells there. And all the little dots that you can see on the screen.
Yeah, ignore these big things.
Those big things are hair. So off skin, especially in animal, you often have hair, a little bit of residual hair that's left, but that's fine. We can we take that out once the skin samples have attached but all the little dots that are on the screen, they're basically skin cells that are starting to attach to the bottom of the flask and begin to grow. So when the flask is covered in, we want it about 50 to 70% covered in the fibroblast cells, which this is a very small one here. We can then harvest those cells and preserve them back down. And that shows that that sample is viable for future regenerative uses, whether that's turning it into sperm and egg cells, which is what Tullis will go on to explain later, or maybe if we want to clone that species to bring back genetic diversity. So that's one element of work using the skin samples, but then we also preserve the reproductive organs. So we'll harvest any available semen, but then we also take the testes and the ovaries and we dissect them and we keep back a selection of that material, with you being able to preserve it and then wake it back up in the future and it being able to remember how to produce semen cells and how to produce egg cells. So you can basically begin to produce lab-grown semen and lab-grown eggs from that animal that's been submitted. So that's what we do for Nature Safe in this room here.
Yeah, I think we've got one more thing, Lucy, that you missed out, because I stole your thunder a bit before. Pam, well, no, we didn't, but what was that animal that you were dissecting the other day? Testicles, so one was related to the elephant.
Oh, yes. You haven't stored it already.
No, we were just saying, explaining.
Yes, so that's a rock hyrax.
Yeah, we were saying that.
It's a rodent that is closely related to an elephant.
Don't worry, we haven't pulled it out.
You've done that without them.
But what I think, can you just explain, because I think this is really special, Kate. I say I've been doing this so long, but I'm always blown away. Every week we seem to be something new that we're divulging. We're talking about the bees this morning, but just I think you should just have a look a bit what's going on here and what project you're looking at, just very briefly. Go on to the next one.
So
fascinating. This is.
We were very lucky to go on a training course to London Zoo and the Natural History Museum for 3 days last month, where we learnt about freezing coral and fish semen. So I don't know how many of you know that coral is actually an animal and not a plant, and it produces sperm and egg cells, and they produce them as a combined bundle, and it releases from the coral stack and floats to the surface of the water, and you can then capture those little bundles of sperm and egg and freeze them down. So we went to this training course and we've come back with a 3D printer because they need a different type of freezing machine to the conventional one that you've seen in the other lab where it's an ice cube one. Instead, we've got a thermos flask, which we're going to print. This is just an empty flask, but inside it, we're going to print a special structure that will hold the vials when we're freezing those coral and fish samples. So that's really interesting. So I have a very good student here at Gemini who has been very kindly persevering with putting together the printer for us. So we're at the stage, it's on and so on. We're just having issues getting the base, the structure to stick to the base at the minute, but we're making good progress with it. So that would be a really nice one to see it grow. And also whether we can, having been on that course, can we actually adapt this to other species as well? Because obviously with zoo animals, a lot of this work will be needing to be done out in the field. So rather than have to think about taking a humongous machine out with them to freeze the sperm, can we think of a novel way of doing it where they could do it in a thermos flask essentially? So that's quite exciting.
Yeah. Thank you, Lucy.
Yeah.
Right, Katie, I'm going to go into the office now and just switch straight out. Did you want a comfort break? It'll take me 2 seconds to switch over, so I'll go straight in there if you want.
Okay, well, if anyone wants to use this opportunity to have a quick comfort break, we will talk amongst ourselves while you move from A to B, Alice. Thank you very much, Lucy, and Team Nature Safe there. It's a fascinating insight into what you're doing, and yeah, amazing that you can preserve coral given the state it's in certain places in the world. That's brilliant.
Yeah, great. Okay, well, I'll log off here and I'll be back in a second.
Perfect. Right. We'll see you in 2 ticks. Thanks, Lucy.
Thank you.
Thanks. Bye.
So I should have said at the beginning, actually, to the audience that we are recording this session. So obviously it will be available to view. All of our webinars are on our YouTube channel, the Dendy YouTube channel. So you can access it at a later date there. If you do have any questions, pop them in the Q&A or the chat boxes and we will try and answer those as we go. I think probably what we will do is Tullis is going to log in and do a PowerPoint presentation. We'll then do some questions with Tullis, and then as he's a very busy person, he can crack on with the rest of his day, and then Andy's very kindly going to do the second half session, and we'll do some questions at the end with him as well. So that's the sort of order of the day, really. So we'll give Tullis a second to perfect. That's coming through brilliantly. Thanks, Tullis.
See that okay?
Yeah, we can hear you and see you perfectly. Thank you.
So lost you, but that doesn't. As long as you can see me, that's the main thing.
We sure can.
Well, not see me. I don't want to see me, but I can see the presentation of it. So that came out totally wrong. That did. No, as long as you see the presentation, that's fine. So thank you. So I'll give you a bit of an insight into what we're doing with restarting AI and transferring over. So I'm going to, yeah, there we go. Go over. So Nature's Safe, as I say, is second year in now. And our idea is to try and save species from extinction by collecting and definitely storing regenerative reproductive cells on that site. So which you've been hearing about. Sorry, I've got to be doom and gloom, but I think it's really important to get across sort of how bad and what place we are at the moment. We're facing our sort of 6th mass extinction at the moment. I think there's about 6 or 7 million species on the planet, 1 million at risk. There's about 40,000 critically endangered. So it's imperative that we obviously do something. And this was a report that came out, I think, about a month or so ago, saying that the wildlife populations declined by sort of nearly 70% in the past 50 years. And that's obviously a huge worry for many different reasons, not just that we're losing amazing species, but for our planet and for us and everything else. So it's imperative that we do obviously something to save these amazing species. And climate change is going up, as we know. The warming's going up. The global is estimated to be sort of 1.5 degrees above what it should have been years ago. So. And what that causes is obviously, as you see on the news, droughts and fires and different extreme weather, which basically loses habitat. If we lose habitat, we lose our species. So it's obviously, this is why we're sort of a race against time, really, to try and biobank as many as we can down. Extinction happens in a normal format, extinction happens, but anyway, normally through natural evolutionary rate, but it's going up nearly 1,000 times more than it should. So we've got to try and slow this rate down as quick as we can. And the time it sort of takes me to do this presentation, what is it? Well, we now sort of an hour or something. 4 more species would have gone extinct, which is so sad. Roughly 1 every 15 minutes, roughly. 50 to 100 species a day. So it's something we have to do really quickly and try and do it for the future. You are the future generations out there. Some of you that are watching this, and you're the ones that are going to have to pick the pieces up. So it's imperative we do something. So cryopreserving, it's called cryoconservation. So our solution, as we heard before, is to preserve these live reproductive and non-reproductive cells as well, so we can bring them back to life. And the different parts of the body that Lucy was explaining about was obviously the testes, the ovaries, and we do the skin. We usually like the ear, just because the ear is one of the fastest regenerating cells in the body. And people say, why skin samples? And you've got to imagine in each skin cell is your unique DNA. And I don't know whether we explain this enough. And so we got the unique DNA, we can do an awful lot more with that DNA. And there's different ways that we can use it, which we'll show a bit later. Don't know, Katie, have you heard of the Millennium Seed Bank? Oh, hello.
Sorry, yes, I have, and as a member of the RHS as well, and all that, they take it very seriously. All these the work on preserving plants, just the same.
Exactly, and there's the Millennium Seed Bank's gone out there and preserved a lot of the different seeds, seed bank, but there's very little on different species, which has amazed us. So we would like to be sort of the equivalent of. And with this, we've had to get, as a big group of expertise come together. Chester Zoo has been one of our foundation zoos, really, that has come together with this, but we've got many other zoos that are sort of really on board with us now, which is great, from all over the country. So it's great to see, you know, this really taking off. As I say, Chester Zoo is the first one, and now we've got ZSL and Patent Zoo and all these other ones, which is great, because without them we couldn't get these samples in here. I don't know whether, Katie, have you heard of EASA? Andy, I'm sure obviously you've heard of EASA. EASA is the sort of the umbrella group of a lot of zoos around Europe, and really there it's really vitally important to get their accreditation. So we got accredited them by the last year to being one of their first living biobanks, and that was a real big step, because without that it was very difficult to go to the zoos and ask them to submit these samples. So so that was an important step. And this next video I'm going to show you is just have to tell me, Kate, if you can't hear it, but hopefully you will be able to. But it's I watched it so many times, and it still puts goose bumps, down my neck, because you see the last two northern whites, but how important it is we do this work.
Hello everybody, my name is Richard Vine, and I'm talking to you from the Old Pejeta Conservancy in Kenya. Behind us you can see the last two remaining northern white rhinos on the planet, and we are doing our best to recover this species from the brink of extinction. Both are infertile, and therefore artificial reproductive techniques are the only options that are open to us. Nature Safe is also playing a critical role in saving the world's biodiversity by creating one of the first living tissue banks on the planet. Their work is incredibly important, and we look forward to working with them in the future.
Yeah, it's a very moving video, that. I mean, why we ever, let it get to that stage. The last two in the world, mother and daughter. So it's incredibly sad, really. So that just demonstrates what work we've got to do. So this is just another diagram just showing how, these different species, how they're declining. You can see the corals is one of the fastest declining of all these different species. And that's, as I say, Lucy was saying that this course that we both went on about 3 weeks ago, I think it was, over 50% of world's coral, has been lost since 1950. So it's one of the most threatened animal species out there. And if we lose our coral, we might well start losing our fish. So it's imperative we do something. This diagram, everything we've spoken to in the last hour or so, is encapsulated in one diagram, really. This is from the Revive and Restore. So what we're trying to do is get these different species, freeze them down, and then by different technologies, which you'll see, it be reproductive gene editing or whatever, stem cell work, or actually mostly the bigger part of it at the end here, not knowing what it's going to be doing, is this is how that can be, use these samples. What we do know, if we don't freeze these samples down now, we are going to lose many of our species. It's imperative. We have got nothing for the future. So that's why we've got to do it here and now. So the science behind it, as I was saying before, I've been doing this for 30 years, cryopreservation, and we take usually the skin samples from quite often the ear when an animal passes away. I think last week there was 8 animals on Wednesday that we did, and it's always very sad because obviously an animal comes in, part of an animal comes in, it means it's passed away. So when you see a few come in, it's an animal's had to pass away to give you those samples. But the beauty about skin samples, you got up to 5 days. So it's not like the sperm or the eggs or the testes where you've got 24 hours or 12 hours. This you've got 5 days. So it's great for moving these samples around the country and things like that to get them to us. So Lucy was saying we do ovarian and testicular samples, and then we preserve them down. We usually do about 10 samples per animal. 4 or 5 remain the property of the zoo, and the other 4 or 5 is we can do trial work. In other words, we just want to grow those cells in a dish, because there's no point just keeping lots of samples if they don't work. You can imagine the task is huge on this. So we have to have a really good data system, and we've got what's called a matcher system, which is these special labels that stick on the vials, cryo vials. So in the tank, we can easily distinguish who they are and find them as well. So there's quite a bit of confusion out there between biobanks and living biobanks. There's lots of biobanks out there, but they quite often store at -80. We store at -196, and quite often the -80 ones are looking at the DNA profiles of those different animals and species, but they can't bring them back to life. So that's where Nature Safe is quite a bit different. We can bring the cells back to life in the future. Yeah, and this is the same thing. That's what the difference is between us and a normal bank, and it's called cryoconservation. We explained a bit like the sucker punch that we saw in the collection area before, but the extinction vortex kicks in when you get these small populations, and this happens a lot on the equine world, and it happens a lot on other species. And actually, it's one thing that usually quite often makes a species go extinct. It's not just the lack of numbers; it's the inbreeding coefficient. And I think, Katie, you were alluding to, I think the Suffolk Punch mares that you had poor fertility, or some of them had poor fertility. And this is what, was that right?
Yeah, one or two of them definitely, and yeah, it was attributed to the sort of inbreeding problems because the lines that they were and number of I mean, this is going back 20 odd years now.
Yeah.
So
well, you're spot on. You're spot on. And the same thing happens with other species out there. So this is where nature, if you've got a genetic line that you can bring back, it can really make the difference between a breed surviving and a species surviving or not. So sort of future technologies out there. And this is where sort of going from sort of, you know, present day to sort of going in the future. Some of the stuff we're doing already with sexing semen, and there's about 4% more DNA in the female sperm, so you can sort of home in on that and pick out those two. They do this with the bovine industry already, and so you know, we're just trying to use these tools now for the equines and other species. So this next video is the foal that I was talking about that was born a few years ago now, but the first one. After 11 months of waiting, Ruby produced a filly foal, and it's not just any old foal. This is a foal born by sex semen. We believe it's the first ever in the world for a rare breed animal to use sex semen. And using these technologies can really help this breed. There's only about 70 or 80 females left in the country. There's only 300 left in the world. They need all the tools of the industry, and just quite incredible now what we can do with science and technologies. And using this technology, maybe, just maybe, we can help stop these breeds from going extinct. But this holds the future. This little girl here. So yeah, it was a long 11 months, I can tell you now, but I've never looked between a horse's back legs so quickly, because there was about a 5% chance it was going to be male, but luckily it was female. So that was a great success story, that. And then again, I know I keep coming back to horses and what can it do, but now this has been transferred onto rhinos, and they're starting to sex rhino sperm as well. So it's incredible where this work is leading to. And the next stage, that was all done with fresh semen. Now we're looking at seeing we can do sex frozen semen. And we had our first pregnancies the other day using sexed frozen ICSI straws, believe it or not. So we talked about the samples. So we get epididymal sperm, testicular, ovarian, or skin. And this is really just sort of infographics showing when we got epididymal, like we did the other day. We can take the semen straight from the epididymis. And we've been doing this again with the equines for about 15 years, but we've done quite a few other species now. Some of it, I say, works. I'd say 50% of the time it works in other species, just purely because we're learning how to process. And quite often when an animal dies at a zoo, it may be through an illness or something like that. So that semen quality might not be very good anyway. So that can be sometimes the other problem associated with that. So this is a cheetah that we did in 2019. And they generally don't have very good semen quality. Again, a bit like Katie was talking about, the inbreeding of the Suffolks, they got the same problem. So, and that's nature's way quite often, but we managed to get some sperm cells and freeze this down. Testicular samples, the tissue, we can actually get the tissue now, freeze that down. And it has been done in mice. We don't do it here, but you can actually culture these and start to produce sperm actually from these tissue samples in the lab, which is incredible. Same thing with the ovarian tissue. This is Professor Susanna Williams is one of our trustees. We can actually grow ovarian tissue, or get the ovarian tissue and culture the oocytes in the lab. And again, we don't do it here, but it has been done in mice. Have we done it in a rhino? No, but it's obviously, we have to look for the future and seeing how some of these samples can be used. And then there's quite a few of these different species that we've used. So this is really pushing the boundaries out now, but it is apparently it is possible to do. It's where you get your skin sample from your ear, like we were talking about before, which got your DNA in it, growing the cell lines that you saw Lucy doing under that microscope, but reprogramming those cells. So at the moment, it thinks it's a skin cell, but reprogramming that cell to neutralize it, to put it into an induced pluripotent stem cell, and then by adding different growth factors, actually turn that into a sperm and an egg, which is quite incredible, because once you can do that, in theory, you can obviously bring back that animal. So it's quite incredible what the technology is. And we've done this, we haven't done that, but we've grown the cell lines, just showing how it works. We froze this particular animal's ear sample down in, I think it's 2019. Yeah, here you go, 2019. And then in 2021, we thawed that ear sample out and grew these cells from that, just to, I suppose, proof of concept to show that this can, how it really works. So we can bring these cells back to life when they've been frozen down for a long period of time. Other sides of it is using skin cells for cloning. The pet and dog market, that's already been done. And the horses, we've got a cloned horse here, we've got a cloned dog here as well. Just shows that this can be done. And we need, unfortunately, or fortunately, depends where you look at it, that domestic side, to really put the money into it. So there has to be a commercial side to do all the research, so we can use this on other species. So this work carried on going, and basically you take the DNA out of that skin cell, take the DNA out of an egg, replace it with the DNA from the skin sample, and by an electrical fusion, they start to simulate and grow. And this happens quite a bit. And this just again a proof, this skin samples taken from a Brzezowski horse 40 years ago. I mean, this effectively, this foal was 40 years old by the time it was born. It's quite incredible how we could bring it back. I think it's a bit of a show jump as well, the way I look at it as well. Right? It's jump in that bucket. But obviously this was an easy win, you could say, because they've done horses before, they've done cats and dogs, and it's with a company called Vargen, who we work with in America. But it just really shows how this can really help. So the next one was to do the black-footed ferret. They virtually went extinct, but you'll hear a bit more about that in a minute. But this was 30 years ago, so and they were so inbred, so it really does work well. Other technologies that are out there is ICSI, where inseminating one sperm cell into one egg, and this actually is a time-lapse camera here, and this was taken every 20 minutes, and this is actually one of the first horse embryos you can see dividing for the first time ever on a camera, which is quite cool. I know it could be any, it looks like any embryo, but it was an equine one at the time. So the sort of the genetic side is really going to be the future, looking at the genetics and genotyping on these different species, and really looking at the genetic architecture of certain traits of interest as well. We can pick them out. I mean, genetics really is the way forward. And this might well blow your mind, this next bit. So this is the future. If you can DNA profile a species, in theory, we freeze it down, you can get the profile. So saying in the future, you can actually bank that, put it on a computer, the profile, and in the future, you saw that 3D printer next door. That was just obviously a one for just producing plastic, but they reckon in the future you'll be able to 3D print the DNA, which is quite incredible. Is it going to happen next year? No, but is it going to happen in the next 5 or 10 years? Maybe. So a lot of these endangered species, they might not be frozen in a tank, although we still need the DNA. They might be stored on a computer, weirdly, and we can 3D print them off. Now, that's really pushing it out there. Sounds very futuristic, but these things can be possible. So yeah, this is very much as a global effort, and we can't save these species on our own. We need your help, and then you've been very, very great at helping, supporting Nature's Safe, and it really does make a big difference. This is my work out in South Africa. It's just my job was I know it sounds a bit weird, but hanging on to an elephant's penis for about 2 hours a day. But we were collecting, we did 8 bull elephants or 7 bull elephants, and it was an amazing experience. But we're using the equine extender to freeze them down. I've just been asked to go out there again to do the rhinos in July next year, which will be fascinating. So a few facts about elephants. There were 10 million elephants at the beginning of the century. I think there's about 200,000 left now. So, and there's some of them where they're starting to get inbred. So it's really important to save the different genetics down. So just so you know, the cost side, it costs about £100 to preserve one animal, which to me doesn't sound a lot. And it costs £5,000 to preserve an entire species, which is incredible. We need 50 different genetic lines from each species to make them sustainable. So this can make a big difference, and in going forward. This is obviously where we're looking for potential funding and things like that. So the future, we're looking to roll this out all over the world. We can't move endangered species across borders, so the idea is to have hubs in different places. And one of our first hubs is actually, it's not going too far, it's going to be in Dublin, believe it or not, hopefully. And then, we are in talks with places in India, Israel, South Africa, and places like that. So that's our vision. And if you didn't know what a mountain chicken frog looked like, this is what it looks like. We've done 137 different species in the last 2 years from 266 animals. So we all think of the rhinos and the elephants, but these other species, a lesser known one, are as important, maybe even more important to the biodiversity and loss of our planet. And what people don't realize, if we lose these species, us as a species will suffer, and as well. And we had the amazing fortunate thing this year, we won the Queen's Award for Enterprise. It was an amazing award that we won this year. And from Stallion AI Services, and the innovation was nature safe. So that was all down to sort of the amazing team that we have here at Stallion AI and Nature Safe as well to get this award. We've been on a lot of press. There's more press going, so we've been very lucky in pushing this out there. We're on BBC the other day. HBO are doing a documentary on it as well. So, and I'd really urge any of you out there that want to get involved, we'd love to hear from you. And it doesn't have to be monetary. It's about opening doors and whatever it may be. Honestly, we'd love to, and it's so rewarding, this. I was just going to leave you this last video, and then I can take some questions with David Attenborough. And I think if David Attenborough says it's got to be done, I think everybody believes it. Yeah, it's got to be done. So this is, he sort of wraps up again what I've been talking about. So thank you for that. Hope you enjoyed that presentation. And there's some of our team who's all doing a run around Chester Zoo to try and raise some money. Hope that was a bit, sort of gave you a bit of an insight into what we do. And it'd be great to answer any questions.
Definitely. Thank you, Tullis. Can I kick off with some questions? I think conservation charities often get criticised sometimes, or the general public do at least, for supporting the fluffy and the cute kind of species. How are you choosing? Because obviously your mountain chicken frog wouldn't fall into that category, so you're not picking on those criteria. But how do you choose which ones you're preserving?
Sadly, it depends which way you look at it, are chosen for us. Basically, when any animal passes away of interest, the zoo send them straight to us. So we can't do anything. We can't do anything on a live animal. So when any time an animal passes away, we bank that and they send it in. So there's a lot of people say, "Oh, I just want to save money to send the rhino or send that," but I said it doesn't quite work like that. We are determined about what the zoo send us for that day and what's passed away preceding the day, a few days before or something like that. So and you did right. If I was to say to you, can you send us £5,000 to save the rhino, I think they'll be inundated with money. If I said, right, can you save us £5,000 to save the mountain chicken? Well, I know people would, but it's just a different analogy to it, and people see it slightly different. But I don't want to put the rhino, because they are amazing, but the mountain chicken frogs is as important as this animal here. So it's how we look at them slightly. We get an array coming through the door.
And it is literally you are open doors to any species from a.
That's right, and even the ones that aren't going, like we did a giraffe the other day that wasn't genetically valuable in a way that we really, but it's really important for us to do the work on that animal, because say we get a very rare genetic giraffe, we don't want to be, I'm not saying we did trials on it, we want to make sure we can grow those skin cells on it, we can save those testes and sperm, and you know, like I said, about 50% time it worked. So the next time we get a giraffe's testicles in, and I can't remember whether it worked or not, it may well have done, but if it didn't, we've got a much better chance of doing the next one. So it's really important that we do them all rather than just the rare ones, because so we can learn, we're basically learning all the time. It's, you know, we haven't got all the answers, but as I say, we're on the phone to somebody in Canada this morning about snake semen and trying to freeze bees. So yeah.
Really important. I'm going to hog the questions unless, Andy, do you have any or
Well, yeah, I do actually.
Go for it. You go first.
Fascinating to hear Tullis's work, because I also used to know Thomas Hildebrandt quite well,
Yes,
who
yes.
you probably know,
Yeah.
who also worked on tapirs and northern white rhinos. In fact, I think did an unsuccessful attempt to do embryo transfer, I think, in one that was in a zoo in the Czech Republic.
Yeah.
So yeah, really fascinating. And I also happen to be a board member of Dublin Zoo, so I pricked up my ears when you mentioned another centre. So
yeah. Oh, very good. Yes. Yeah. Brilliant. Andrew, Mooney. Yes, that's it. Yes, that's right. Yeah.
Yeah, he's the conservation officer there, and I actually chair their conservation and research committee. And their ethics committee actually as well. But so
it's great to see. Sorry we were to sort of hog about Dublin Zoo, but it is so important to Nature Safe, because that has been the first one, because we've been trying to roll this out. I went to a conference in Portugal the other day, which was the EASA conference. There was 950 delegates. So I'm not a zoo person myself, but going in there, it was quite an eye-opening experience. And we came away from there absolutely mind blown how people, we did a presentation to them, and they just loved the concept. And Andrew was there, and so many people came up to us and said, God, we want to set up a Nature Safe in Israel or South. We said, look, Andrew, they're setting up the first one. We really want to, I'm not saying use you as the sort of the template, the guinea pig one, but you ought to be the first one that we're going to use, because you're closer, it's easier to do. And he was so enthusiastic about what this was going to do. And so it helps us, so we can get a blueprint how to set them up, and then obviously we can roll this out around the world. So no, well, thank you for being part of that, because that makes a big difference to us.
Yeah, I mean, it's basically been approved by the board, so I think you probably know that anyway, so I'm not telling any secret.
But as long as you know, it really does. For us, trying to work with a new, but miles and miles away is a little bit more difficult, but in Dublin to set up that first one is just fantastic. It really is. So.
I think some other questions that I would have, Tullis, in terms of what I'm fascinated by is the animals you're taking from a zoo domestic environment versus wild. Do you see any difference? Because you'd imagine the nutrition of the domestic animal would be better, so perhaps semen quality would be better, or I don't know. Does that stack up, or actually is the domestic environment not as good as the wild environment?
Oh God, I'll be careful how I answer this question, because I'm not into the zoo. But we do speak to Chester Zoo, and we work very closely with Chester Zoo, and they look at the environment that we have our sort of stallions in, and we're learning all the time from our stallions and how we look after them. And what we do know is how you look after your stallions and how you feed them plays a massive part in what the semen quality, their reproductive side. I know there's lots of others, the health side and everything else. I suppose I look at the reproductive side, and it makes a huge difference. I mean, it really does. And I suppose it's a side that we're all learning more about, and especially the zoo side are learning more about actually feeding and doing certain things. And stress is one of the biggest players, not the biggest, but one of the biggest players in poor semen quality. And sometimes with these, equines we have here, you can't even see their stress, but it's because they don't show that stress in a different way that we expected. But we do notice it sometimes with the box walking or anything like that. And if we can change that, that has an effect. That simple thing can have an effect on semen quality. So yes, environment, feed, where they are, all has an effect on fertility. And you hear some species, you could say a bit like the mountain chicken frog, we've found really difficult to breed in captivity. So yeah, so they're all different, but then there's so many species, as we know, crikey, without the zoos, they just wouldn't be alive. And that's what people have really got to understand as well, because, you know, it's so important, you know, the work they're doing out there, you know, where they've got fantastic breeding programs. And now they're learning about the genetics, trying to get the right genetic matches to stop that in that sort of extinction vortex, which is absolutely vital.
Yeah, and I think the other increasing threat of disease and new diseases coming into our shores is, particularly for our rare breed species, is really, really important. And a great example, we worked with Balmoral stud. I think Balmoral Lord came down to you to be collected from, and sadly, I think it was 12 or 18 months later, succumbed to grass sickness. So you know, having his semen now available means so much to obviously the stud manager and Her Majesty, before she sadly died earlier this year, but prior to that, it made a big sort of difference to accepting the loss of that stallion, knowing his genetic material was conserved for the future.
We see this so many times that it's so sad when an animal passes away and they haven't banked its genetic material, and it is so easy, even if it's a tissue sample, it's very straightforward. Just have to admit, sperm and eggs are a little bit more difficult. But you're right, there's a valuable lesson to learn. Lucky they did, but what happens if they haven't done with that animal? No. Yeah. And the Highlands, you know, there again, they're on the endangered list. So,
yeah, very much so. And I think, just to reiterate the point you made with the Przowski's horse, one of the things that really alerted me to the potential benefits of these gene banks is in the rare breed context. And people were talking about being able to access a Hereford bull from 50 years ago, and just how much we've moved the breed type away from what it perhaps started as. And you know, sometimes breeding becomes so intensive for a particular fad or trend in a domestic species, and we take it in a direction that isn't actually sustainable long term, or it moves it away from what its original traits should have been. And it allows you to try and go back and create an animal that's more true to type, you know, maybe from 50 years ago. So I know in the rare breeds world that is particularly valuable, because sometimes we'll get a bit carried away with these small populations. And
so true, and we've seen it with the Cleveland Bays and things like that, and now they're actually doing really well. Their inbreeding coefficient has gone right down, which is fantastic. So it shows it can really work.
Yes.
So, but no, I think we never know when we're going to use these samples, and the black-footed ferret and the Przowski horse are classic examples how they've been used in a positive way.
Yeah, definitely. And I guess one last plug, I suppose, for what we're doing and why this is so important to us is, as a feed industry, you know, you showed those pictures of those species that are being ever sort of encroached upon by human activity. And so for us, it is really important not to be contributing to palm oil and destruction of rainforests and things like that around the world. So the low feed miles, you know, is a key to us. It's a really important selling point. I probably think we don't make enough of it, to be perfectly honest, because, you know, it's our activities around the world collectively that are destroying this habitat for these amazing animals, and it's something we feel very, very strongly about.
Well, I really appreciate your support. I really do.
Well, I wish we could do more, to be perfectly honest, but it's a little drop in the ocean, I'm sure. But we'll
it's great to meet Andy as well. I mean, that's I have to admit, naively, I didn't know the link with Dublin Zoo. I can't tell you how excited we are, Andy, to have.
Well, that's great, Tullis. I don't know, Katie, if you saw the question Tracy put up about careers. Perhaps you could just take that and ask Tullis.
Yes, definitely. So we have got some students listening, and I think if they wanted a career in this area, I'm not sure where they would start, but have you got some advice, Tullis, apart from coming to do work experience with you, I guess?
on the career side, is it? Yeah. It's so we have a lot of work experience students. I must admit, I think we have 20 or 30 through the year, and it's so I think, yes, it's definitely, to me, I would recommend it wholeheartedly. I've done a full circle. I was into the, obviously, as you know, the horse breeding, and now I think 80% of my time is now into the conservation. It is the most rewarding thing you can do, is to do this work. It's just fascinating. I do it every day of the week. So I would highly recommend it. Obviously, you've got to earn a living out of it, and that's really important. So so it depends which side you wanted to go on. From our side, yes, we do welcome people here. We are obviously very limited how many people we can take. Obviously, we only take 1 or 2 students on at a time. But yeah, we're more than happy to hear from you. But yeah, on the zoo side, I have to hold my hands up. Andy might be better than me to answer that. I don't know how you get into the zoo world. to me, but also can know in the conservation world. And if I always say, if I was to come back again, I would be a vet and go and do the work out, because I'd go out to South Africa, because I'm not a veterinarian myself, but I'd love to do that work. I'd always like to come back and do that work with, do more of it full time in conservation, using cryopreservation, because it's just fascinating. And we're always learning. I mean, being on the call this morning about freezing bee semen and snake semen, where's that going to go? It's fascinating. So, yeah.
Yeah, never a dull moment, that's for sure.
Yeah.
Brilliant. Okay, well, I think we'll wrap up there with your part of the presentation. Thank you ever so much, Tullis, and Team Nature Safe behind the scenes there. India doing a sterling job behind the camera, and Lucy and co in front of it as well. So thank you very much indeed. I will introduce Andy. So I've got his, very kindly, his sort of biography and career details here. So, and I think it is important just to share it, because there is so much experience that we're going to be privileged to hear from him. He has a lifelong association with animal production and management, having graduated with an honours degree in agriculture from the University of London. He started as a lecturer at Sparsholt in Hampshire, where in 2002, in association with the British and Irish Association of Zoos and Aquariums, he devised and submitted the curriculum of what is now the Diploma in the Management of Zoo and Aquarium Animals to City and Guilds, which is widely recognised now as the gold standard for training zookeepers. Since then, he's worked for the Royal Zoological Society of Scotland as their nutritionist in residence, and during this time he oversaw the bespoke formulations of feeds for grazers and browsers, which have widely adopted in the UK and European zoos and wildlife parks. In his time at Sparsholt, Andy was also responsible for initiating a 5-year nutritional advisory contract with a large safari park in Guangzhou in southeast China, and consulting on the nutrition of a wide range of species. And in 2014, he was conferred the honorary title of Director of Zoo Management Studies, which takes him all around the world to give talks at zoos. Andy's research interests are currently focusing on diets for ungulate herbivores, in particular facilitating the feeding of browse alternatives, which leads nicely into the presentation, and additionally reviews of nutritional composition of eucalyptus for koalas and analysis of bamboo leaves and shoots for giant pandas. I think this covers the highlights of a very extensive career and demonstrates why I'm delighted that Andy can actually join us this afternoon. So if you're happy to share your screen, Andy, it would be great to hear more from you.
Thank you.
Thank you very much indeed. All working okay? Oh, brilliant. There we go.
Hope everyone can see that. So I have one apology, actually, which is I haven't got the dengue
oh, don't worry.
logo on there, Katie, actually, but I would like to acknowledge the Zoological Society of Hertfordshire, or Paradise Wildlife Park, who do support now, some of my work. Now I'm more freelance. So a very warm welcome to everyone. Thank you for your interest in joining us this afternoon. So Katie and Tracy have asked me to talk about a long-term interest I've had, which is really in using Lucerne or Medicago sativa, to give it its posh Latin name. As Katie said, many people defer to the American terminology of alfalfa, but I kind of like Lucerne to show my British roots. So one of the sort of the introductory slide I'd just like to show you is this one, which illustrates why certain forages are actually not suitable for all types of herbivores. It shows there at the bottom that grasses are one type of plant and non-grasses are another. Often grasses are called monocotyledons because they produce one seed leaf in germination, and the non-grasses are dicotyledons because they produce two. And actually they have, in terms of their plant composition and structure, they're very different. So to actually assume that all herbivores can eat grass is actually somewhat of a misnomer. Quite a few of them are specialist foragers, and actually eating grass is to their detriment. So I'm going to say a little bit more about that later in some of the other slides, but just in case you've never taken in the beauty of this plant in full bloom, here is an image of a lucerne field in about July time, I would guess. And you can see the close-up of the flower, and it's interesting that Tullis was talking about bee semen, because obviously bees are important pollinators of this type of crop, albeit we're not really interested in the seeds. It's actually in the foliar material. So what particular species are we really interested in that are these niche foragers? They're actually called browsers, and I ought to explain really that a browser is a herbivore that doesn't focus on grass. It browses leaf material from trees, bushes, shrubs, and forbs, as they're called in Africa. So I just wanted to show you a few of these species in their sort of physical appearance, so you can appreciate that there are a number of them. On the left-hand side of the screen, we've got the elk, or the moose, we call it. The Americans call it an elk. On the right-hand side is an okapi, which is actually similar to a giraffe in some respects, although it's not biologically related. But they're very specialist browsers found in the Democratic Republic of Congo, and are, at risk from the numerous wars and things that are going on out there, civil wars. On the left of this one, we've got the giant eland, sometimes called the Derby's eland, after Lord Derby, who discovered it in Africa. And then, of course, there's the very well-known giraffe, showing basically this browsing technique in captivity on a feeding pole. Then here we've got a greater kudu, or a giant kudu, on the left, and a bongo on the right. And here there is a gerenuk, which again is quite an endangered species. It's one of the most tricky ungulates to keep in captivity, and it has this browsing habit where it stands on its hind legs, as you can see. Actually, on my screen, it's sort of excluded by the gallery view, but I hope you can see at least the hind legs on the ground, and the nose up higher. So what is actually true of a lot of these browsers is that they have what's called a prehensile tongue, and this is an okapi reaching up into the leaf canopy just to browse. So the tongue is about 18 inches long, in fact, in its entirety, but they are able to use this tongue to wrap around the twigs or the foliage, and then pull it into the mouth. So this is an unfortunate okapi that was autopsied in a post-mortem lab at a veterinary centre, and I was fortunate enough to go along and see it. And you can see on the left-hand screen, for instance, that the tongue is basically, it is a very long, extendable organ. So in their native environment, this group of herbivores or browsers actually, as we said, basically eat non-grass materials. And here's a picture of some acacia on the left that I took when I was in South Africa a few years ago. And it basically is very different in its composition, to grass, as we're going to see in a moment. So the challenge in captivity is really trying to imitate as closely as we can the diet in a non-native or non-native habitat, actually in a captive environment, because clearly trying to grow some of these species is not straightforward. They're not frost tolerant. They don't like low temperatures. So we have to try to find an alternative solution, which is non-native species. And here again is another close-up of these things, which you can see have particularly vicious thorns. And these are thought to be a natural defense mechanism of the plant, so that actually a browser can't just go along and strip the whole lot off. It basically can get its muzzle between adjacent thorns and actually browse selectively and then move on. So in captivity, it says browse in UK zoos because that's where I mostly have worked, as Katie has said. So this is a plantation, a browse plantation at a wildlife park in the northeast of the country, and it's actually a standing of willow. So willow is often chosen as a browse species, mainly because it's very easy to grow, grows quickly. If any of you have ever come across coppicing for fuel production, willow is often used because it grows rapidly. It takes 6 to 9 months to establish, and after that you can get a reasonable crop every year, and you can coppice it, and in other words cut it off, and then it shoots from below that cut, as you can see all along here. So zoos are now planting willow, not only willow, hazel, beech, other species, but they tend to take longer to grow and to establish, so their harvesting potential isn't quite as great as willow. And one zoo in particular has about 60 acres now, or 25 hectares of willow planted for feeding a variety of browsers. So why lucerne? Well, basically we come back to this as a plant that is entirely suitable for feeding this rather fastidious or picky group of animals. Lucerne is a legume, as Katie said. It's a dicotyledonous plant. The structure of the leaf and the stem is much more akin to browse, which is also a dicotyledonous in the dicotyledonous group. So lucerne is, relatively easy to grow, and we understand its requirements quite well. So one of the key features is its protein level. As a fresh plant, probably you would expect to get protein levels of 18%, certainly 16%. I've even measured values as high as 20% in the past. And when you think of what a grass is, if you get 10 to 12% in a grass, you're doing well. And that only happens early on in the grazing season. So as we said, this nitrogen fixation is very sustainable because it does it through what's called symbiosis, as you may have learned in biology, where the nodules contain these bacteria, rhizobia, and the rhizobia basically harness the nitrogen from the atmosphere. And they can fix appreciable quantities of nitrogen every year. But the key thing, I think, as well, and that's what I'd like to stress, is not only is the protein content important, but the fiber fraction is actually more entirely suitable to these creatures, and it is much more digestible. There are also additional spin-offs in that they have a very good profile, particularly in magnesium, which, and in fact, they're probably one of the better sources of magnesium in any forage crop, and certainly higher than grass. And one of the reasons for this is that they have a deep taproot, which enables them to withstand extended periods of drought. But that deep taproot also enables them to bring up minerals that are held lower in the soil profile. They also include tannins, which are an interesting group of compounds because some people regard tannins as being anti-digestants. They particularly bind with proteins, but they do also have potential as natural wormers in animals. And there's been quite, some a revision of interest in this, because if you can basically feed a plant and it provides you with this spin-off, it's actually well worth having. And another plant called sainfoin is particularly good at preventing bloat. It has higher levels of tannins than lucerne does, but there is also thought to be some anti-bloat property in lucerne, although it can also, lucerne can cause what's called legume or frothy bloat. So tannins, again, have some scope in being useful, certainly in stopping the production of methane, which is one of the major greenhouse gases you may hear about on the news. So I want really now just to show you in fairly graphic terms why this difference in plant type is important. This is the skull of a giraffe. It's not particularly pretty, and I sort of apologize for shocking you this late on a Wednesday afternoon, but this was an animal from a safari park that I got involved in some while ago, and you can see the lower teeth on this jaw, the incisor teeth, are in very poor condition. The reason being that this animal actually had been fed on grass, hay, not exclusively, but predominantly. So what has now appeared in the top left-hand side is a picture of what giraffe teeth should look like. And okay, these were taken in a young male. And this animal that you're seeing a picture of was probably over 20 years old, the one with the grotty teeth. But this actually is how the teeth should look. And I think you can see quite clearly that there's a big difference between these teeth and the other ones. And so what's happened is that basically these teeth have worn down because of the silica content of grass hay, and this is part of the difference in the composition. Grass has quite a high silica content. It's very tough. It's quite abrasive, and obviously, as an animal eats and chews the cud, this silica causes very rapid erosion of the teeth. This doesn't happen, in fact, when the animal is fed browse, or in our particular case, lucerne, because lucerne doesn't have silica as an abrasive material, which is again another reason why it is so important. The other thing just to say is that grazing animals basically have a different type of teeth. They have much higher ridges than the browsers, and they have these very definite crescents of dentine, which is quite a hard material, and so they can cope with the abrasive nature of the grass, whereas the browsers can't. So basically, in this tooth wear, there are two things. One is silica or sand, and there's another material called phytoliths, which are also made up of silica. And so this is what is important in causing this excessive dental erosion. And equally, when animals graze close, grass and close to the soil, they inevitably pick up silica, and intentionally or unintentionally, and this actually helps or accelerates this erosion process. So there's tooth-to-tooth contact when animals chew, but diet abrasiveness is a very key factor. And this again sort of illustrates the difference between the teeth of grazers on the right and browsers on the left. You can see that a browser has a very low crown, whereas the grazer has a very high crown with defined surfaces. So this is quite a busy slide, but what it's actually trying to show in the three ringed numbers, the red ring numbers, is the acid detergent insoluble ash is what ADIA stands for. In other words, this is basically inorganic material of which silica is a major component. And you can see that the values for grass and grass hay are actually much higher than lucerne, probably 3 times as much in grass as lucerne, and when the material is dried, it goes up to almost 7 times as much. So, and this was a study that was done in rabbits, but it sort of can be extrapolated to other species. Rabbits, of course, are well-known grazers in their habit. But so this underlines the message that lucerne has much lower levels of silica and abrasive material in it. And as we're talking to an audience who are particularly interested in equids, you can see, though, that even in wild and zoo equids, I think this is a zebra, that the dentition of the wild animal, as shown here, is much more uniform. Here's a picture of a zebra in a zoo, and you can start to see that there is uneven growth on the tooth, and you have these sort of very sharp areas, which again has been probably due to inappropriate feeding and not supplying enough grass fiber or abrasive material. So the other thing just to say, and I won't get too involved in this because it's quite a story, but a lot of the herbivores that I'm talking about, these browsing herbivores, are also ruminants. And this is basically a stylized diagram of the digestive apparatus of a ruminant. So you can see it has this huge fermentation vat called a rumen, and then various other associated structures. This is actually what it looks like for real. This was a giraffe that was euthanized, unfortunately, because of very severe arthritis. But you can see that here's the rumen, bang in front of your eyes, and it is quite a large structure. When you open it out, you can see basically that this is actually for an okapi rather than a giraffe, but this is actually what the fresh rumen looks like on the left. On the right is actually the same organ, but with all the food removed and the surface actually enlarged. So you can see that the rumen consists of all these thousands of papillae, or little finger-like structures, which are very important in absorbing some of the products of digestion. So basically, and I say this is perhaps a little bit over the top, but it explains how rumination goes, that the animal actually eats material and then subsequently it will regurgitate it, chew it again, and then basically that reduces the physical size, and then the bacteria and the protozoa, which are actually in the rumen, can digest it. So really, the whole of this function is actually feeding a population of squatters that actually live inside the animal and do a function that the animal itself can't do. So if you've ever watched a cow or a giraffe or any other animal chewing the cud, you can see the little boli or balls come up and down the neck in a sort of muscular ripple. As a teenager, I used to watch this with quite a lot of interest, but probably explains a lot about my strange personality. So I want now just to come back to this issue with plant structure and explain a little more detail about the difference between the two, because here we've got a grass, and then you can see in the middle a closer or more magnified picture of that grass leaf, showing that you've got a midrib in the middle and then straight lines. And if you actually project that to the right, each cell in that grass structure is needle-like, long and thin. Whereas in a non-grass species, the plant's fiber, and this is the crucial thing, is much more multi-sided or polygonal. So you can see the very clear difference in those two fiber structures. And that's important because if you try to feed the needle-like thing to a browser, all that happens is that it stays in the rumen a long time and basically is much more resistant to digestion rather than the browse or the non-grass material. And as I say, this is a crucial difference. The differences in digestive strategy mean that you shouldn't ever contemplate really feeding a browser grass because of the way that it behaves in the rumen. So food passes through the rumen of a browser much more quickly because it really needs a more digestible food source than a grazer. So the posh word for this is that browsers have lower rumen particle retention time, which is a posh way of saying that food passes through their gut pretty quickly. And this is, again, a stylized diagram to show the difference between a browsing rumen and a grazing rumen. In the grazer at the bottom, it eats large amounts of grass, forms this layered or stratified approach where you've got fibrous material on the top, and then you've got, well, actually, you've got gas on the top, then a fibrous layer, and then you've got liquid and small particles which are being digested. Whereas in the browser, basically you have a layer of gas on the top, but then everything else underneath is a much more uniform or homogeneous mix, as it's known. And this is basically the crucial difference. And it comes down to this difference between browse or non-grass food and the grass material. Similar thing, which again, I don't really have too much time to go into this, so I'm going to gloss over it. But it's an interesting slide in that it actually shows for a non-grass species that basically the leaves have quite high protein, shown up here, and they also have in the stems a reasonable amount of protein and actually quite a lot of fiber. Here as shown by the NDF or the nutrient fiber of the cell wall. But the important thing is that even though there's a higher fiber, it's quite digestible. And this again is a bit of a boffin diagram, but it shows that as the plant ages here, so from vegetated into flowering, the digestibility, i.e. what the animal can use in the material, plummets pretty quickly. And so really you want to try to feed material which is pre-flowering if you can. Incidentally, as the plant ages, the protein content goes down as well, but the cell wall or the structural carbohydrate goes up. So it's really a balancing act between trying to get all those parameters right. So coming to the important bit of the talk really, which is how do we use lucerne for these zoo animals? So we can use it as haylage, which is basically where it's cut and allowed to wilt. We can cut it as hay, where it basically spends about 5 to 6 days out in the swath. If the weather's good, you certainly couldn't make hay with the kind of weather we've got at the moment, neither would you. Or you can actually feed it as pellets, and again, if you're well tuned to Denji's product profile, you'll know that they actually sell and market lucerne in this form, as well as in the Alpha A, which is a dried form. Both of those are entirely suitable. Quite a few zoos do use haylage. The problem is that it's like grass silage. It's basically quite moist, so it needs to be kept in a sealed bag. And if the bag has a hole in it, you get secondary fermentation occurring. So it's quite an unstable food material. The other thing that you need to have quite a lot of mouths to eat it, because once you cut the bag, and you need to feed it out very quickly, because within a day or so it starts to secondarily ferment. So this is the difference really between the two. On the left we have a sample of lucerne hay, and on the right a sample of the haylage, which is actually much more leafier. The other thing I put in, since I last did this presentation, I put in an extra few slides following some photographs I discovered when I used to work in the Middle East in the United Arab Emirates. This is actually lucerne hay from America, basically in a bale, which is known as a single press. And then, so you can see that basically the leaves are quite fully formed and the stems are identifiable. This is what's called a double press, where the material is actually really subjected to a large amount of pressure in a ram, mainly so that it's much smaller to transport. But you can see the huge detrimental effect. It creates a huge amount of dust when you actually try to open this. Basically, you need a mask on it. It just is so dusty. So that double pressing has really ruined a lot of the feed value, and it's why how you store and transport the product is actually really important. So again, this is just a comparative picture showing the two. So coming to, again, some more figures, and I know that it's quite bad just to sort of look at a screen like that and you think, well, wow, you know, what's all this mean? Basically, the important tables are actually the ones, the column, the second column in showing the pellet composition, and what we can't, or what I can't quite see, is the values on the right for grass hay. But if you can see them, you'll see that the lucerne products generally tend to have a much higher protein content. They might have a higher fiber content, but the fiber is digestible. They also have, as I say, a very favorable magnesium content, 0.2% as opposed to 0.08% over there. So the feeding value of lucerne for these animals, as I say, is actually quite important. Now I'm going to rush through these because this is actually just showing you a little bit about the operation of how you harvest it, which is perhaps not entirely relevant to most of you. But basically, this is a standing crop actually being mowed, and you can see on the right hand side of the picture the swath. This is more of the same, and then there's a mechanized operation to collect it either by a baler, as is the case here, or a forage harvester, which I don't have a picture of. And so the bale is left in the field like this. If it's hay, it can then be picked up and transported back to a barn, or if it's haylage, it can actually be wrapped in black plastic. And that's actually what's happened here. So you can see the material needs to be conserved and preserved from exposure to the air. So slight digression here, but I wanted to show you a particular study of where, you know, lucerne has helped a safari park that I've worked with for quite a while. They have an old a female who has teeth that look like this. They are very curved or concave. Normally an elephant's teeth should be flat. This was actually an animal from Dublin Zoo that photographed a few years ago. So the teeth should sit like that, not like this poor lady here. I think she's about 30 years old, so she's survived well to now. She actually had feces because she couldn't chew her food. Basically, the poo was coming out in sort of 2-foot lengths, which is highly unnatural. So as a result of this problem, we've actually introduced chopped lucerne into her diet, alpha A in fact, and it's now restored her gut to normal activity, and she doesn't produce these rather long poos. The other thing which I'm quite interested in is a relative of lucerne called sainfoin, which basically is, again, quite indigenous to the UK. In fact, before the Second World War, it was a major part of forage production in the UK, but lost out big time to some of the ryegrasses and some of the other plants which establish quicker and produce higher yields. But one of the things about sainfoin is that it's actually quite suited to chalky soils and porous soils, which we have in the south. So on a porous soil situation is probably a better alternative to lucerne, although perhaps it doesn't yield quite as well. It also has a higher tannin content, and so therefore is a better protectant against bloat. And like lucerne, though, it's rich in protein. And as with lucerne, bees love fertilizing the flowers. And again, it has some advantages that it flowers long from May till October. So from a harvester forage point of view, means you have a longer window, but obviously the later you harvest it, the lower the food value is. So I'm just going to, we've mentioned tannins, and I don't want to dwell on this, so I'm just going to quickly spin through this. Yeah, as I say, I think it has a niche, but it's certainly lucerne and sainfoin are very good. So to just sort of sum up before I talk about one last other exciting development, why is lucerne a good food for zoo browsers? As you've hopefully picked up the message, it's the best alternative which is available to natural browse. And a lot of browse plants, lose their leaves. So unless you can actually conserve browse by drying it, which is exactly the same problem as we have with other materials, then it's not going to give you 12 months supply, whereas lucerne is available all year round. We've talked about the plant structure and the fiber being much better. It also ferments very well in the browser's rumen, and it has a very good profile of minerals and protein. And as we've shown you, there's flexibility of use. Denji produced both pellets and dried lucerne as alpha A, so it's really good. So in the last couple of minutes, I just want to talk about something that Katie and I have been working on recently, and indeed Tracy, who introduced me to this product when I was visiting, called Alpha Beet, which is actually this unique combination of lucerne and sugar beet pulp. And basically, this is actually quite an exciting product if you're a zoo nutritionist, because it means that you've got really good protein from the lucerne, you've got really good fiber from the sugar beet, and it's the right sort of fiber. It's like lucerne, it's non-grass fiber, so really suited to rumen fermentation in a browser. It's also has all the credentials of being an ideal hard food for zoo browsers. Those of us who've been in the field a long time have had quite an issue getting traditional feed compounders to actually make a browse hard food. They love to put cereals in it when we really don't want cereals, because those are high in starches and sugars, which are rapidly fermentable, and we don't really want that. And the lovely thing about Alpha Beet is it is highly palatable. If you know anything about beet pulp or speedy beet or any of those products, generally they have a very small residue left after the extraction process. So we know it works, thanks to the generosity of Denji. We're trialing it in a couple of zoos. This is a shot from Blair Drummond Safari Park in Scotland, very nicely showing the food bag and a curious group of greater kudu. And actually it's gone down really well. This is a close-up picture of a greater kudu female actually tucking into Alpha Beet. And finally, just to sort of say about its composition, again, I'm grateful to Tracy, I think, for this data on the Alpha Beet composition. And you can see it's got, 14% protein, which is pretty much where we want it to be. It's got good fiber characteristics. It's got low starch and low sugar, which again are very desirable from a browsing fermentation point of view, and really brilliant calcium to phosphorus ratio. I mean, that's amazing, and really good magnesium again. And it compares, I've just put here a couple of things to compare it with. Vitality Browser, which is one of the products that Katie mentioned I was responsible for helping to formulate, which is now a big product in the zoo world. And then you can see Alpha A just on the right hand side, the other side of it. So Alpha Beet compares very well with a commercial browsing pellet, and it certainly compares very well with the Alpha A as well. They're very complementary to each other. So last slide, the traditional one. Thank you for listening and your long suffering. And if there are any questions, I'm sure we can do our best to answer them. So I'm going to stop the sharing there, Katie, and hopefully that then brings it back to you. Oh, there's Tullis still with her.
Yes, and a special guest, I think.
Yes.
Who's that, Tullis?
Yes, this is Gemini, so she's a very special guest. She's actually a clone dog.
Right.
It shows that.
So she's older than she looks then.
Yes, that's right.
That was brilliant, Andy. Thank you ever so much. Can I ask, I should share the fact that because we're on Essex clay, we have struggled trying to grow samphoy, and it does feature in our sort of insect borders and things. We're helping pollinators, but as a crop, you alluded to the low yielding, and it certainly doesn't tolerate the Essex clay very well, which is a shame, because in the era of anthelmintic resistance, which is a really hot topic in the veterinary profession at the moment, its natural properties in that area could be really quite useful. But have you used it much with the zoo?
Yeah, yes, we have a little bit, Katie. I've got nothing to report on worm egg count reductions yet, but we are using it in a safari park where they're feeding it to giraffes, and their veterinary team are on board to actually start trying to do comparative worm egg counts from feeding sainfoin and not feeding it, but nothing concrete as yet, I'm afraid. So no, I think the point you make is right, that it's not okay for clay and heavy soils, but most of Hampshire is chalk downland and very poor grade 3, agricultural land. And that seems to be where it might have more of a niche, where, Lucerne might not actually do quite so well. So I think, there might be, niches where it works.
Yeah. Absolutely, and I think it could be a really good break crop for farmers as well.
I've actually got going some in some of my raised beds this year for porpoises next year. So I want to see how stop them eating the flowers and give them sandfoin instead.
Lure them away.
Yeah.
Diversionary tactics. The other question I had, we'll see if any come in on the chat as well, but obviously with sort of horses and things, we don't have actual probiotics, live bacterial species approved for horses, but do you have any sort of experience of using things like prebiotics or yeast for zoo animals, or do you indeed have a probiotic you're allowed to use?
Yeah, I mean, I think it's an interesting one. I don't think they're used as a matter of course, but they are used, you know, as a strategy for encouraging animals back to health who've had digestive upsets. So there are a range of prebiotics, certainly sorry, probiotics that are used, but they're not used routinely, I don't think.
No, it's interesting to know what what bacterial species. Has anyone sort of profiled any of the stomach contents or ruminant contents or anything?
No, not as far as I know. Unless it's been done in the last couple of years. I mean, we did actually do a lot on giant panda gut profiling. We did a study with Aberdeen University actually a few years ago, but not actually in browsers as far.
Well, one would imagine, I guess, that it's going to be similar.
Well, I don't know, actually. I mean, I think that's the interesting thing. I think actually if you profiled the microbia of grazing guts as opposed to browsing, you would find quite a difference. I mean, you know, we tend to talk in generic terms about, you know, the holotricha and the microorganisms that actually are free living and float around, but I don't think there's been so much work done on the bacterial side of things. And of course, fungi is another ball game altogether, because you've got, you know, the combination of the protozoa, the fungi, and the bacteria all subsisting in this wonderful foul-smelling soup that is the gut contents of these animals.
Yes, I think it's similar in terms of fibre digesting being the dominant in a healthy one, and if as soon as we start to put these starchy cereals in, probably tipping it into a more dysbiotic kind of state. But yeah, it would be fascinating to look, wouldn't it, and see just how it compares, if we could ever get to the point of trialing what the natural environment would be, and then the domestic sort of zoo environment.
Oh yeah, absolutely. I think that's right, but I'm not aware of. I mean, there used to be a team at Reading University who were doing a bit, but I'm not sure whether they're even in existence, because like everything else, they've been subject to, you know, to cuts and funding issues. But
yes, and is that, well, if you had some unlimited budget, what would be an area of interest, a hot topic at the moment? Is there anything you would love to be researching if money?
Well, I mean, I think the whole issue of, you know, the candidate species in these digestive organisms that are fulfilling the role, you know, of the fermentation of browse and lucerne, and, you know, whether they are different or similar to those that occur in you know, grass fermenters. Really, I think everyone assumes that there's similarity, but I think I'm not sure whether that assumption is based on any hard fact.
Yes, and similarly, from the question the students are still watching, what would be your advice for starting a career as a sort of zoo nutritionist? Obviously, you started, I think, in agriculture, ended up towards zoo. Are there courses now specifically for zoo nutrition? I imagine not, but you never know.
No, that's a really interesting thing. I mean, I often describe myself as a recycled farmer, in that I grew up in the agricultural industry, and that's why I went, you know, to university. There weren't all these wonderful, you know, degrees in animal science and animal management and zoo stuff that when I was a teenager. So really, I kind of ended up by accident doing what I'm doing, in that I always had an interest in anatomy. I did that as my special electives at university in digestive physiology. in the second year, and that's really where I sort of got to interface with the rumen. They actually had a fistulated cow on the farm where I was, and so that was the sort of icing on the cake in this digestive physiology module, to go and stick your hand in the rumen of a cow, which was, well, it's an interesting experience. But yeah, so I mean, I think really, if anyone is interested in this, and I do get asked sometimes, what should they do? I do recommend, a life sciences degree, whether it's in conservation biology, animal management, that sort of thing. You mentioned also the thing about work placements and what have you, and obviously that's good, because it's really, how do you stand out as a candidate from everyone else who wants to do the same thing? So you've got to really, I think, put yourself out there, do as much as you can, volunteer like there's no tomorrow, and hope that when you're in the position that, when you leave your qualification, that you can, either get into a zoo or an animal collection or whatever. But it is hard, actually, really hard. And I only ended up, as I say, by accident doing this, because I started running zoo management courses in the late 1990s because of a demand, and then eventually ended up running this scheme for all 92 zoos in the UK and Ireland, and we went international to Dubai and Riga. So, we were delivering it not quite all over the world, but, distance learning is a huge thing these days, and we pioneered a lot of that. So yeah, it's really just, I think, don't specialize too soon, is my advice to anybody. Do a broad-based degree or qualification, and then look to specialize once you've actually graduated, and don't give up. I mean, that's probably my best bit of advice. Don't, you've got to be prepared to take bruises and knocks, and just get up and keep going if you really want to do that. It is as simple as that, I think.
Excellent advice, I think. And just one last question for me. How are zoos, because obviously you're very close to zoos and you're on the board of Dublin and things, how are they catered for in terms of veterinary? Because I guess exotics is not something that's covered very much in the curriculum, as is nutrition generally, but specifically zoo animals.
That's a very interesting question, Katie, because when I first started getting associated with zoos in the 1990s, very few zoos had their own vet. They would call on the services of a local practice and then buy in specialists. Pretty much now, every zoo has its own vet. And there's been a great proliferation of vets. I mean, they breed incredibly well. They're not endangered species like
Tullis then.
No. I mean, if you get one vet, you then need a second vet because you have to cover the holidays. And then, vets proliferate like mad. And I must be careful in case there are any vets listening. But
I've shared this with lots of vet students. So
yeah. So it's actually really interesting. I mean, the amount of nutrition advice and study that vets get is quite limited. I mean, I guess lecture at Glasgow University, and I know from their curriculum that they might get, about 6 hours of nutrition in the whole 5 years, which is not really a lot. No. So yeah, I think vets and nutritionists can work together and should work together, but it doesn't always quite work like that. It depends on personality, of course. But yes, so zoo vets often are expected to cover nutrition. And I found this myself when I was at Edinburgh. There were 4 zoo vets there, plus me, and we used to have a nutrition focus group and discuss nutrition and diets and everything. And both parties contributed. So I think it's there are things that nutritionists can learn from vets, and things that vets can learn from nutritionists. So.
Definitely, it's always a collaboration, and I think it's even broader than just their interest in nutrition. I'm always curious because, again, to come into a zoo environment having probably had very little sort of coverage of exotic species, full stop, health, veterinary technique, all that kind of thing. But I guess if you've got someone experienced there, you bring the juniors in and they learn on the job, so to speak, handling these animals.
That's right. Yeah, absolutely. And the other thing, just to say, actually, I'm also sit on the European Nutrition Group, which is part of EASA. So there are now people coming together to prepare reports and references and guidelines and all the rest of it on how you feed different taxa.
Yeah, no, that sounds great. And I don't know, Tullis, whether you have any thoughts on that. How's your interaction on the veterinary side with fertility and zoos, and are you collaborating with them?
On the veterinary side, you're saying?
Yes, yeah. Is it? I guess you're able to impart your knowledge to them, really.
Yeah, I'm on the reproduction management group as well, which is got a lot of veterinarians from the zoos all around, and it's I feel like I only had a tiny little bit, but it is fascinating on that side of it. Yeah, but we're just obviously adding our bit on the reproductive world and new technologies and how it can help. What's been great is really trying to get the vets on board, because without the vets we couldn't.
No, exactly.
Veterinarians that are doing the post-mortems, the veterinarians that are obviously taking the samples. So it's really important for us to engage with them at the zoos. So it's absolutely vital. They're our key, really. Yes, we have to get the MOUs and memorandum of understanding signed with the zoos, but actually it's the veterinarians that the key part to Nature Safe to get the samples to us. Because some of them are very sort of, they worked really heavily. Sometimes they've got a big workload, and this just adds to it. So
yeah. No, it's a subject very close to my heart in terms of collaboration between sort of allied professionals and all these things that you're just joining up everybody. And again, it's just sharing of expertise and knowledge, and amazing how much you learn from one another just through ad hoc conversations. And suddenly you take a leap forward in sort of knowledge and understanding.
So true. Yeah, and that's what we had this morning.
Yeah. We haven't had any questions, and I think that's probably a great point at which to say thank you both very much indeed, and is one of the reasons for wanting to do a webinar such as this. And I'm glad you've got to meet as well.
Yeah, I know.
It sounds like
that has. It was me talking about Dublin Zoo.
Yeah, well, there may be some overlap in future, which is brilliant. And I'd just like to thank you both very much, and Tullis behind the scenes as well, your team there. Andy, thank you for your support. We really appreciate everything you do, and setting up these trials for the alpha beats just been very exciting for us, because it takes us into new territories as well, which is fantastic.
Well, I'm actually going to the other zoo tomorrow.
Brilliant.
Katie, so I'll see how things are going there. Well, I think it's actually not arriving till next week, but I'll make sure everything's in place.
It's much appreciated, and Tullis, phenomenal, amazing work that you do, and thank you on behalf of everyone that's listening. Really appreciate
Thank you for your support. Thank you.
Thank you. Well, I think we have to end it for everyone, so I'll say goodbye, and just if anyone does have any questions at a later date, if they send them in to us, we can forward them on, or as I say, the recording will be on our YouTube channel in due course. But thank you both very much, and have a lovely evening, everyone. Take care then.
Thank you.
Thank you, Katie. Thank you. Thanks, friend.